Hello there, ladies and gentlemen. My name is Mr. Williams, and in this video today, we're going to talk about meiosis. Now, right away, if you see that term, you're thinking, sounds a lot like mitosis, and it is. Very, very close. But it's a completely different type of cellular division, and it's got a completely different type of result, and an entirely different purpose. So without further ado, let's go ahead and jump into meiosis. Meiosis is a form of cell division that halves the number of chromosomes when forming specialized reproductive cells. Those specialized reproductive cells being gametes or spores. Meiosis involves two divisions of the nucleus, unlike its brethren mitosis, which is just one division of the nucleus. We get really complicated when we title the two divisions of the nucleus for meiosis. We call the first division meiosis 1, and we call the second division meiosis 2. Just like with mitosis, we've got similar stages of prophase, metaphase, anaphase, and telophase. Okay? We will have prophase 1, metaphase 1, anaphase 1, telophase 1. And then we'll have prophase 2, metaphase 2, anaphase 2, telophase 2. So meiosis 1 and meiosis 2, the two stages of meiosis. The generation of reproductive cells, the generation of gametes, the cells that are going to hold the information that's given from one male, one female, fused together, boom, the directions to build a brand new organism. Okay, we have some of the basics for meiosis out of the way. Let's go ahead and jump into the details for each one of the stages for meiosis 1 and meiosis 2. Prophase 1 is when we start to see the chromosomes condensing and the dissolving of the nuclear envelope. Homologous chromosomes pair up along their length and then carry out a process known as crossing over. Crossing over assists in giving us great genetic variation between some of these chromosomes. You've got like areas on like chromosomes that are detaching and reattaching at the other like areas. So they're kind of swapping certain segments of their DNA between one another. After prophase 1 is complete and the crossing over has occurred, we see the lining up of these homologous pairs towards the equator of the cell. The pairs of homologous chromosomes are moved by the spindle fibers to the equator of the cell, and those homologous chromosomes, each made up of two chromatids, remain together. Okay, so the pair is actually what's going to go ahead and be pulled apart. All right, the actual homologous chromosome structure itself remains intact. Independent assortment takes place kind of between metaphase 1 and anaphase 1, where there's really no rhyme or reason to how these chromosomes and these homologous pairs line up on the equator. A whole slew of possibilities for independent assortment here when we get to the metaphase and anaphase stages of both meiosis 1 and meiosis 2. Anaphase 1, similar to mitosis, we've got the actual pulling action, the actual separating of some of these entities towards the equator of the cell. The homologous chromosomes separate, so the pair has been torn apart. As in mitosis, the chromosomes of each pair are pulled to opposite poles of the cell by the spindle fibers. But in meiosis, the chromatids do not separate at the centromeres. Okay? So kind of sharing a little bit of like vocabulary here, the word chromosomes getting thrown around a lot. But in review, we've got homologous pairs lined up at the equator, and those pairs are split. So homologous chromosomes are now moving over to the opposite sides of the cell, to opposite poles of the cell. Telophase 1 is next, and that's when the individual chromosomes gather at each of the poles, and in most organisms, the cytoplasm divides, forming two new cells. So that does it for meiosis 1. Okay, meiosis 2. Prophase, metaphase, anaphase, telophase, all with the Roman numeral 2 besides it. In prophase 2, we've got a new spindle that forms around the chromosomes. Okay, we actually didn't have any unwinding and unzipping. We just have the chromosomes still intact. Okay, in metaphase 2, we see the chromosomes line up along the equator again, and they're again having the spindle fibers attached to the centromeres. So we've actually got the homologous chromosomes lining up along the equator, and now the spindle fibers attached to the centromeres of those homologous chromosomes. We move on to anaphase 2, and that's when we have the centromeres actually divide, and now we've got the chromatids being pulled to opposite poles of the cells. Those chromatids are now moving to the opposite poles of the cells, and once they arrive there, telophase 2 takes place, and that's when we have a nuclear envelope form around each of the sets of brand new chromosomes. Okay, so those chromatids, once they're over there by themselves, we start calling them chromosomes again. The spindle breaks down and the cell undergoes cytokinesis. Cytokinesis being the actual splitting of the cellular cytoplasm. The result of meiosis then is 4 haploid cells. So half the amount of DNA that you would originally start off with if you were referring to an human organism. That does it for meiosis 2. For a little historical perspective and some visual aid, we're going to go ahead and check out a little clip here from Bill Nye's 100 Greatest Discoveries in Biology. Take it away, Bill.
Around the time that Walter Fleming was studying the cell division process, biologists already knew that fertilization resulted from the union of 2 sex cells, sperm and egg. What they didn't know was why these were the only types of cells in the body capable of reproducing new life. What made them different? The first answer was provided in 1883 by Belgian zoologist Edward van Beneden. Van Beneden was studying a species of roundworm when he discovered that nearly all of its cells contained 4 chromosomes. The only exceptions were the roundworm's sex cells, the male sperm and the female egg. These cells had only 2 chromosomes. Van Beneden observed that once 2 of the sex cells came together, a full set of 4 chromosomes appeared in the fertilized egg. But just why the sex cells had only half the number of chromosomes compared to all the other cells was still a mystery. Then in 1887, German biologist August Weismann entered the picture.
He knew about the work of Van Beneden, who had shown that the sperm and the egg bring in equal numbers of chromosomes. But Van Beneden's work had not really worked out the details of how the chromosomes were reduced in number.
He saw them go in half, and he saw them recombine.
He saw that the sperm and the egg brought in half the number of chromosomes as the body cell, but he didn't really know how they got rid of the half that they got rid of. So Weismann discussed how this might happen from a theoretical standpoint. And he said, well, what you could have would be that either every one of the chromosomes would just split in half, and half would go off one way and half would go another way, or you could have it more complicated, that the chromosomes could pair, and then instead of splitting, they just separate again.
After years of studying the behavior of chromosomes, Weismann determined that at some point a developing organism signals its sex cells to divide their number of chromosomes in half. Weismann called this very special form of cell division meiosis.
I think they will look back and see this as the golden age of biology, because, let's say, 200 years between 1800 and 2000, we went from not knowing what distinguished a stone from a frog, okay? And now we know the difference between a stone and a frog.
That's pretty good.
So that, in a real sense, we've answered the old philosophical question of what is life. Maybe not to everybody's satisfaction, but we're well on the way.
Okay, so after our initial discussion over meiosis, and after more discussion involving the specific stages of meiosis, and what is happening in each one of the stages, in each one of the phases, okay, and after our video here from Mr. Bill and I, giving us a little historical and alternative visual perspective over the meiotic process, let's go ahead and take a couple of minutes here to kind of look at it big picture, because we're just jumping into this complex patterns of inheritance unit. All right, and this is kind of the initial process that causes some of the information that we are going to look at later on to be altered, or to be mutated, or to be adjusted. All right, or to cause some issues, maybe good and or bad, down the road. So that's why we're looking at meiosis right now. So let's kind of take a little bit of a bigger picture approach to some of this material, and it's going to deal a lot with just reproduction in general. Some organisms look exactly like their parents and siblings. Others share traits with family members but are not identical to them. Some of the organisms have two parents, while others could have one. Depends on what you're looking at there. The type of reproduction that produces an organism determines how similar that organism is to its parents and to its siblings. Reproduction, the process of producing offspring, can be asexual or sexual. So that reproduction word, we've already seen that before when we looked at mitosis, making more cells. And we've also seen it when we looked at meiosis, making more sex cells. Okay? So, two types of reproduction, asexual and sexual. Kicking it off with asexual here, it's when we have a single parent that passes on copies of all of its genes to each of its offspring. There's no fusion or haploid cells being fused together, such as gametes. An individual produced by an asexual reproduction is actually a clone, okay? So it's an organism that's genetically identical to its parent. As you read through some of your texts and as you read through some of the other sources throughout this unit, prokaryotes are ones that typically reproduce by asexual reproduction, and it's asexual reproduction known as binary fission. There are also some eukaryotes, though, that produce asexually. Typically, your single-celled eukaryotic organisms. In contrast, sexual reproduction is when we've got two parents and each form reproductive cells. Those cells have 1/2 the number of chromosomes that actual organism possesses. A diploid mother and father would give rise to haploid gametes. We just saw that process with meiosis. Okay? Those haploid gametes are then going to join to form diploid organisms. Because both parents contribute genetic material, the offspring have traits of both ma and pa, of both parents. But they're not exactly identical to each one of the parents. They're half of the male, half of the female. Sexual reproduction with the formation of haploid cells occurs in eukaryotic organisms, especially human beings. Okay, that does it for this video. In review, hopefully you've got a grasp over what the process of meiosis is. Okay, really similar to mitosis. We've just got some special stages, special phases, some special little adjustments to a few of those stages and phases that you need to be comfortable with. Also, be comfortable with the terms crossing over and independent assortment and how they can assist with increasing the genetic variation that could be present in some of the haploid gamete sex cells. Okay? Be able to identify some of the stages and some of the structures that are present in meiosis. And finally, have a little bit of information down about asexual and sexual reproduction. Okay? The two ways that organisms can actually make more of themselves. That does it for this video. Hopefully it's helped you out a little bit. If not, watch it again. And by all means, ask any questions that you may have. Thanks for tuning in.